next speaker is Tuncay from Turkey.
Hello everyone. Today my presentation will be about the wound healing potentials of 3 Achillea species on cultured NIH3T3 fibroblasts. I'm from Turkey. My name is Tuncay, Ağar. I'm going to talk about these titles following order. First of all, Asteraceae family. I will give a small information about the family and the genus Achillea. Also, I will talk about the purpose of this study. These are my materials and methods. Before I introduce the wound healing potentials, I will talk about the preliminary screening studies, determination of wound healing activity, and I will finalize my speech with the conclusion part. Okay. Asteraceae family. The alternate name is composite, you know. The Asteraceae, with its approximately 1,620 genera and more than 23,600 species, it is the largest family in the world wide. For my country, Turkish flora, it is the second largest family. If you wonder about the first one is Orchidaceae. Anyway, if I talk about the botanical features of this family, we can say the calyx of the flores may be absent, but when the present is always modified to pappus. For inflorescence, we say it's in a head or in capitulum, and there are usually 5 stamens in this family, and if we can say syngenesis stamens. That means the filaments are fused to the corolla, and while the anthers generally connate. This is the syngenesis anthers that looks in this family. And botanical features of my plant, Achillea. Achillea is one of the famous plants which belongs to this family, Asteraceae family, and 54 taxa are indigenous to Turkey, and of which 31 are endemic. Achillea is perennial pilose herbs, often with thick, woody rootstocks. Leaves entire to 3 or 4 pinnate per teeth and alternate. Capitula usually arranged in a terminal corymb. Flowers white or sometimes yellow. Ray flowers are female. Disc flowers hermaphrodite, regularly 5-toothed, and pappus is absent for Achillea. Due to numerous medicinal properties, Achillea species commonly known as yarrow had been used in folk medicines for thousands of years and worldwide. We can say in Turkey, various species of the genus are used in wound healing, for abdominal pain, for stomachache, symptomatic relief of colds, against ulcer, diarrhea, as diuretic, emmenagogue, appetizer, carminative, and insecticide agents, and lots of usage for this plant. And similar ethnobotanical uses and some veterinary uses for some species were also reported for many experimental results, for many articles. Besides the medical applications, some Achillea species were also consumed as vegetable, beverage, spice, additives in food and cosmetic products, and horticulture. Today, some therapeutic applications such as anti-inflammatory, wound healing, antispasmodic, hepatoprotective effects are proved, their activity proved. Previous phytochemical studies on Achillea species have revealed that genus is rich in terpenoids and phenolics as possible bioactive compounds. As flowered plants, Achillea species produce essential oils and flavonoids, lignans, amino acid derivatives, fatty acids, alkamides were reported from the genus. And what is the aim of our study? In the study, we have evaluated the wound healing effects on NIH3T3 fibroblast cells of 3 Achillea species growing in Turkey. The name of the plants: Achillea kotschyi, subspecies kotschyi, Achillea coarctata, and Achillea lycaonica. I will use some abbreviations for these plants: AK, AC, and AL. Let me continue. Materials and methods. The plants were collected during the flowering stage from different provinces of Inner Anatolia. The collected samples were identified and the washer specimens were deposited in the herbarium of Hacettepe University, Faculty of Pharmacy. And collection sites and herbarium codes of the plants are given follows. And this is the map for the collection sites. Now I'll talk about the extraction procedure. The aerial parts of plants were dried in shade at room temperature and ground in a mechanic grinder to fine powder and weighed accurately. The powdered plant material were extracted with methanol for 3 days. Methanol extract of each plant material was filtered through regular filter paper and evaporated in reduced pressure until dryness to give crude extract. Before we do the wound healing activity, we made some preliminary screening. These are my preliminary searches: total phenolic contents, HPLC analysis of phenolic acids, DPPH radical scavenging activity, and beta-carotene linoleic acid assay. Total phenolic content. First, I want to say the yields of my extracts as shown in the slide: AC, and for AK, and for AL. I used Folin-Schicalto colorimetric method for total phenolic content to measure total phenolic content. And significantly, the highest total phenolic content was 148 milligram gallic acid, 2 gram extract in AK, Achillea kotschyi. For HPLC analysis of phenolic acids, determination of phenolic acids was performed using a reversed phase HPLC system. Standard phenolic acids were, as shown, gallic acid, protic, and so on, with finished T-cinnamic acid, 12 of these kind of acids. And we use propylparaben for internal as an internal standard. These are chromatograms. First, this is the for AC, and below you are these compounds are the below the chart, which we found for phenolic acids, and for AK we found that chromatogram and that compounds, phenolic acids. For AL, Achillea lycaonica, the identified compounds as shown below. Yes, all phenolic acids as shown in table except T-cinnamic acid were detected and quantified in all Achillea extracts employed in this study. T-cinnamic acid was not detected in any of the extracts. On the other hand, vanillic acid was the most abundant phenolic acid in AK and AL. You shown that. While caffeic acid was found in the largest amount in AC for Achillea coarctata. Radical scavenging activity. In this study, free radical scavenging activity of the extracts were evaluated by DPPH method in comparison with that of a synthetic antioxidant BHT at different concentrations. Results were given as inhibition percent, which is a parameter widely used to measure the free radical scavenging activity. The highest radical scavenging activity was observed in AK in the plant Achillea kotschyi with 26.60% inhibition, while AC showed the lowest radical scavenging property with 6.10% inhibition. Beta-carotene linoleic acid assay. The beta-carotene bleaching method is based on the loss of the yellow color of beta-carotene due to its reaction with radicals which are formed by linoleic acid oxidation in an emulsion. The rate of beta-carotene bleaching can be slowed down in the presence of antioxidants. The decrease in absorbance of beta-carotene in the extracts of Achillea and well-known antioxidant BHT, which was used as a standard. In this test system, it was obvious that AL oxidized most rapidly with percent 81.47 inhibition. The antioxidant capacity of the extracts by beta-carotene linoleic acid system decreased in the following order: AL, AC, and AK. And this is the chart table for total phenolic content and antioxidant activity of Achillea extracts. This is a summarize of the of my speech for total phenolic, DPPH, and beta-carotene linoleic acid assay. And now, determination of wound healing activity. Wound healing activities were evaluated by comparing with titrated centella extract, centella asiatica extract named TECA, on NIH3T3 mouse fibroblast cells, which are proposed as a method for testing wound healing activity in vitro. The NIH3T3 mouse fibroblast cells were grown DMEM medium, dulbecoous MEM medium, supplemented with 2 mm L-glutamine and 10% PBS and 1% penicillin streptomycin at the temperature of 37 degrees in humidified incubator with 5% carbon dioxide atmosphere. TECA and AC, AK, and AL extracts were dissolved in DMSO at a Stokes solution, and the Stokes solution was diluted to the required concentrations. The same dose of DMSO at 2 microliters in 1 milliliter was used for the control and experimental groups and compared with both DMSO and blank controls. The cell monolayers were grown on cover slips. A total of 70–80% confluent cells were treated with TECA and AC, AK, and AL extracts, and these are the concentrations as shown below. Staining and morphometric analysis of culture NIH3T3 fibroblasts. HD15-1 kit Mason's trichome stain kit were used for the staining of cultured the fibroblast cells. After staining, the controls and extract treated fibroblasts were examined under a light microscope with a fully automated photographed attachment using 400 times magnification. Fusiform, polygonal, round, and vacuole containing fibroblast cells under mitosis and the total cell numbers and collagen granules in cell that are counted areas. For my statistical analysis, we used statistically programmed software SPSS, and statistical differences between the experimental groups were determined by one-way analysis of variance, means ANOVA, and Tuncay's test in SPSS. And differences were considered significant if the shown below. And these are the results and discussion. How can we determine our results? Fibroblasts in a polygonal shape are experimental measure for migration, while fusiform fibroblasts are accepted as quiescent cells. Vacuole-containing cells are a measure for aging of the cell population and cytotoxicity, while round fibroblasts are the cells which lose their viabilities, excluding the dye. Total cell numbers and cell numbers under mitosis were used as parameters of proliferation of fibroblasts. And this is a demonstration for this. For example, these are the mitosis cells, polygonal shaped, fusiform, granule containing cell, vacuole containing cell, and round cells. I will summarize all of them in a table. Morphometric analysis performed in this study showed that there were no significant difference between the medium control and the MSO controls you see in the chart table. In addition, the DMSO concentration used here was found to induce no significant negative effects on cultured the fibroblast cells. Significant increases in total numbers were found with TECA at the concentrations of 2.5, 10 microgram when compared with both medium and DMSO controls. Among the Achillea extracts employed in this study, total cell numbers increased significantly only in AK at the concentrations of 5, 10, and 20 for AK. TECA also exhibited increases. Let me show the quick way. These are the, you see, from BP for AK. This is significant. And for mitosis, you see for control group and AK 2.5 significant, and the fifth one 5 microgram, 10, 20, and here with the MSO controls there are significant here, and for 5 microgram, 10 microgram, 20, and finished. The percentage of fusiform cells significantly decreased. Before that, I'll show the same. There is no negative effect for the MSO and controls, not changing too much. And again, we can talk about the significance between the groups. For fusiform cells, you see from group and for AC 40, and with AL 40, from the MSO control with TECA, our control group, AC K for AL here, and with our group TECA shown significant activity 2.5 microgram, 5, and for AC 20 for AL. And I will quickly past, you see the significance between the groups. Let's show others. Significance between them. And for polygonal cells, you see the significance with TECA in these concentrations, with AC in these concentrations, and for AL in this concentration. And for the MSO controls, you see the, again, significations between groups. And the last table, again, there is no negative effects. For round cells, you see the significance between groups. For vacuole containing cells, you see the groups. If you want, I can show you in coffee break, because it's a long table. There are lots of significance here. I'm passing quickly. Here, vacuole containing cells for AL, for the MSO control between AK, and for number of granules, again I show the significance in table. For conclusion, I can say we have determined the phenolic content, phenolic acid composition, and evaluated antioxidant, the wound healing effects of NIH-3T3 fibroblasts of Achillea coarctata, Achillea kotschyi, subspecies kotschyi, and Achillea lycaonica. Achillea kotschyi was found to be the richest species in terms of total phenolic content. DPPH radical scavenging assay indicated that Achillea kotschyi extract had the most powerful radical scavenging property related with its phenolic contents. According to comparative HPLC study, which showed qualitative and quantitative differences in phenolic acids, the most abundant phenolic acid was vanillic acid in Achillea kotschyi subspecies kotschyi. Among the Achillea extracts employed in this study, only AK, Achillea kotschyi, increased mitotic activity and total number of fibroblast cells in various concentrations that indicates possible effects of the plants in the proliferation phase of wound healing. Furthermore, a decrease in the percentage of fusiform cells and a slight increase in the polygonal fibroblasts observed in the study showed that AK also stimulates the migration of fibroblasts to close wounds. As an indicator of non-cytotoxic effect of AK, round fibroblasts were found to be slightly decreased, while percentage of vacuole containing cells decreased significantly. AK also stimulate collagen production in fibroblast cells at low concentrations. In this study, we suggest that AK, because of its antioxidant potency and prominent wound healing property, may be considered a source that could be pharmaceutically exploited. And these are my references. Thank you very much for your kind attention.
So, is it very hard to count the cells under the microscope?
It is hard to count because there are lots of areas you have to count and not photograph from. You have to count from only microscope, because when you taking a photo and if you said I will count with the photographs, it's not good way, because three diagonal dimension is changing. When you're using with micrometer with microscope, you can see the cells. It's hard and you have a headache when you are counting. But if you,
Accurate.
sorry,
Is it accurate?
accurate,
Accurate.
accurate.
Accurate. So the method.
Sorry.
So you're sure about your counting the right number?
Yeah. For right numbers, I'm not only counting by myself. Also, I'll show my advisor. We are making together. Yes. Thank you.
Thank you very much.
Thank you very much.